Today's topic is Topoisomerase. It is a nuclease that breaks a phosphodiester bond in a DNA strand. Hi, I am Manti, and in this video I am going to discuss about this topoisomerase and its types. The first topoisomerase was discovered by James Wang in 1971 from E. coli. There are several types of topoisomerases present in eukaryotes as well as in prokaryotes. All these isomerases can be classified into two classes, that is type 1 and type 2, depending on whether they can clip one or two strands of DNA. Here, type 1 topoisomerase clip one DNA strand and pass other strand through the break before resealing it, while type 2 topoisomerase clip both the strand and pass another double strand through the break, followed by resealing of the double strand of the DNA. Type 1 does not require ATP for activity. The reaction is driven by the energy stored in the supercoiled DNA. So far, the only exception is reverse gyrase, which introduce positive supercoils into the aid of ATP hydrolysis, whereas type 2 topoisomerase do not require external source of energy for the cleavage and relegation during reaction, but they do utilize ATP hydrolysis to drive conformation changes in the protein during the reaction cycle. Now, let's discuss about one by one. First of all, I will start with type 1. Now, about type 1 topoisomerases. It is operated by forming a transient phosphotyrosine covalent bond with one end of the broken DNA strand, either the 5' or the 3' end, which is then followed by the passage of the unbroken strand through the break and ultimately resealing the break. These topoisomerases can be further divided into two subfamilies, that is type 1A and type 1B. Type 1A binds covalently with 5' phosphate, whereas type 1B binds covalently with 3' phosphate. Type 1A topoisomerases passes a single-stranded DNA segment through a transient break in a second single DNA strand, whereas type 1B topoisomerases breaks one DNA strand and allow one duplex end to rotate with respect to other, which surrounds the remaining phosphodiester bond. Here in the figure you can see type 1A topoisomerases affect topological changes in DNA through a strand passage mechanism, whereas type 1B affect supercoil relaxation by breaking a single strand of double-stranded DNA, and it allows one DNA strand end to rotate with respect to other around the intact phosphodiester bond on the opposite strand. Topoisomerase 1A comprises three distinct phases, that is eubacterial topo 1A, then eubacterial and eukaryotic topo 3, and then eubacterial and archaeal reverse gyrase. These enzymes are primarily responsible for relaxing positively and negatively supercoiled DNA, except for reverse gyrase, whereas type 1B appear to be represented by a single family member, that is Topo 1B. Next, about type 2 topoisomerases. It makes a transient double-strand break in the DNA helix and form a covalent lineage to both the strands of DNA helix, and it occurs at the same time. During catalyzes, the enzyme introduces a double-strand break in one DNA strand, which is termed as G segment. and it passes a second DNA segment which is termed as T segment. Here, thus it is the general mechanism of type 2 topoisomerase that it can break both the strand of double-stranded DNA and it can pass another double-stranded DNA through the transient break. And this enzyme can bind to and bend the G segment. After that, the binding of the 2 ATP molecule allows the opening of the DNA gate and passes the T segment. The T segment is dissociated from the top of the enzyme, which leads to unidirectional strand passage. This topoisomerase 2 has two subfamilies and they are type 2A and type 2B. Type 2A is found in all cellular organisms and is again divided into three classes, that is eukaryotic topoisomerase 2, that is topo 2, and then bacterial topoisomerase 4, which is denoted as topo 4, and bacterial and archaeal DNA guides. Whereas topoisomerase 2B include topo 6, which is found in plant and is homologous to SPO11 present in Saccharomyces cerevisiae. Both the type, that is type 2A and type 2B, use a duplex DNA for passage mechanism and have the same ATPase and cleavage domain but differ in overall tertiary structure. Both the type 1 and type 2 topoisomerase change the linking number of DNA. Type 1A topoisomerase change the linking number by 1 and type 1B by any integer, while type 2A and type 2B change the linking number by 2. So this is all about topoisomerase and its types and its subtypes. Thank you for watching this video.